Should we get started, do you think? Okay. Hi, everyone. Welcome back. Um, it's good to see that many people came back. That's a good sign. So, yeah. So, this is the last session of today's meeting. And today we're going to conclude on health effects and regulation, which kind of go hand in hand as well. So, we're very excited to have an excellent group of speakers today. So, I'll be going over some of the health effects just in a bit. But I also wanted to briefly introduce our speakers. We have Amy Pedula from UCSF, which will talk to us about PFAS and reproductive endpoints as a potential regulatory endpoint. Dr. Leila Chatsi as well from University of Southern California. We'll talk about PFAS and NAFLD and obesity. And then we're going to move on to regulatory standards with Matthew Small from US EPA Region 9, as well as Christopher Banks from OIHA as part of Cal EPA, talking about regulation of PFAS at the federal level, but also here in California as well. So with that, I'll get started on the health endpoints. And I wanted to kind of give a brief overview of some of the things that have been the most reproducible, at least in the epidemiological literature and some of the work that we've been doing as well. There we go. There we go. Took a little bit. So we have been chatting about how PFAS are so persistent in the environment. They're also very persistent in human fluids or in the blood, which are probably the main medium where you can find them. Anywhere between 3 to 8 years for the long chain PFAS, or what we now call kind of legacy PFAS. You know, some of these studies are coming from occupational studies of humans working in occupational settings, where it has been estimated for PFOA and PFOS. Half-lives ranging anywhere between 3 to 8 years. They don't really metabolize. Then again, I'm talking about the legacy PFAS in the body, or very slowly. So they're mostly excreted through feces or through the liver very slowly. So this is why they have such a long half-lives. So you combine these two things, very continuous exposures because they're persistent in the environment. Even at low levels can lead to very high levels in the human body because they're not clearing up very fast. So with exposure, the main exposure route being drinking water, this is a major issue. As you can imagine, things can bioaccumulate fairly quickly. And there's widespread contamination. This is just one stats, but the Swedish Chemical Agency, I believe in the European Union, identified over 3,000 PFAS that have been registered. I believe the EPA has that number around 4,000, but as you know, there's many more thousands being identified. So I do want to mention that I think policy works, and I wanted to show some maybe hopeful data. These are some of the legacy PFAS. You have PFOS there in green, and PFOA, PFHX, and PFNA. And the levels of both PFOS and PFOA have decreased dramatically. So this is data from the National Nutrition Examination Survey, or NHANES, which is a representative sample of the US population. And it's done cross-sectional across many years. And you can see that after 3M phased out the production of PFOS voluntarily in 2002, levels have dramatically decreased thereafter over a decade. The same thing with PFOA, where about 6 of the major producers of PFOA voluntarily phased it out in 2006, followed by decreasing levels in the plasma at the population levels, at least in the US, as well as some of these other PFAS as well. But this is a very small chemical space, as we've been talking. There's many more, some of them who have been increasing because of production or replacements. Okay, so what about human health, right? So IARC, which is the International Agency for Research on Cancer, as part of the WHO, recently, last year, completed their hazard identification. And it is important to note that IARC doesn't do risk estimates, so they either classify things as carcinogenic or non-carcinogenic. They evaluated 2 PFAS, 2 of the most commonly detected ones, at least in humans, PFOA and PFOS. PFOA was classified as a group 1 known human carcinogen, and this is based on evidence for cancer in animals. Some things like hepatocellular carcinoma or adenoma, as well as pancreatic cell adenoma as well. What's interesting is that most of the literature was on Sprague Dawley rats. So these are experimental rats, and most of them were males. There were some suggestions for female rats, but the evidence was a little bit more limited. PFOA was also classified as a group 1 based on mechanistic evidence, mostly two specific epigenetic endpoints, the DNA methylation and microRNA, and that came from human studies as well as an experimental model system. The other endpoint that is shared between PFOA and PFOS is immunosuppression, and that has been very consistent across multiple epidemiological studies. Where documented vaccine titers decreases have been seen with both prenatal and postnatal childhood exposures. PFOS didn't reach that level of classification, so it's now classified as a group 2B, as a possibly human carcinogen because of the lack of animal data, but there was mechanistic data in terms of epigenetic dysregulation and immunosuppression, particularly the decreased vaccine titers that has been seen at least in some of the human studies. I also wanted to bring to all of us, to our attention, vulnerable populations, right? So we're thinking about our own exposures, but what about regulating for people that are most at risk? So these plots show PFOS and PFOA levels measured in a birth cohort. So this is from age 0, from birth up to 60 months. And you can see here that breastfeeding duration is a major exposure route, and that has been documented across other cohorts as well, that infants that are exclusively breastfed have higher levels of PFAS. So that tends to peak during the first year of life, where mother and infant pairs are exclusively doing breastfeeding. And when you compare that to formula-fed infants, the levels are much, much lower compared to breastfeeding. Of course, breastfeeding is a very good thing, but paradoxically, that we have to keep in mind that that's a major exposure route. And these levels are much higher compared to adulthood. So data that exists kind of shows that these levels tend to go down during childhood after breastfeeding duration, and the environmental exposures are not as high as what people or infants are exposed to breastfeeding. So it can go, the estimates are as much as 30% increase in PFOS or PFOA per month of breastfeeding. So we have done some work looking at PFAS as endocrine disruptors. So a lot of the work that has been done has been around cancer, but of course we're interested as potential endocrine disruptors. That might be one of the pathways that leads to cancer. So we, in a prospective cohort, these are Kaplan-Meier curves people follow for over 17 years. We documented that PFOA specifically led to an increase in diabetes risk. A doubling PFOA was associated with a 14% risk increase in diabetes. We nested this within a randomized trial, and interestingly enough, the panel A on the left are individuals that were randomized to placebo, and panel B are individuals that were randomized to a lifestyle intervention of exercise and diet. And what we're able to see is that individuals that are adhering to a very strong regimen of exercise and diet, the association is attenuated, so it's not present. It's only individuals that were randomized to the placebo. So there might be an interaction with lifestyle factors as well in terms of the exposures, and that might explain some of the heterogeneity that we see in the epidemiological studies. So that's something to keep in mind about differential response in human populations, and thinking about Western diets and Western lifestyles as well, which might put some of the developing countries at a higher risk of PFAS-related health effects. I also want to bring attention to the need for high-quality prospective studies. So this is a meta-analysis evaluating multiple studies. On the left, I don't mean for you to read these forest plots, but just to show you that on the left are case control studies and cross-sectional studies of diabetes, measuring PFAS and diabetes prevalence at the same time. You can see the first two panels, you actually get protective hazard ratios, which are counterintuitive to what we found and hypothesized associations. And then when you actually, and this is just comparing people that are highly exposed to non-exposed, then the studies that are actually doing proper exposure assessment, measuring levels, are actually showing pretty null associations. And these are cross-sectional or case control studies. However, when the meta-analysis separated the prospective studies, so these are high-quality prospective epidemiological studies, when you measure exposure and you follow people over time, we see very consistently that both PFOA and PFOS are risk factors. The estimate, the meta-analysis risk estimate is on the positive direction, although there's not that many studies, but this suggests that proper study design and exposure assessment are very important to develop proper risk estimates at least. in the same study where we follow people of close to 17 years, we have seen increases in weight or weight gain as well. You can see that the lifestyle intervention is the group that dropped quite a bit of weight. So that's 10 kilograms, which is very drastic weight loss. The effects of PFAS were not evident in that group. And then, however, the placebo group, who dropped a little bit of weight, I think it was on average about half of a kilogram because they were given advice on diet and exercise, but not true intervention. There was an association with PFAS levels and weight gain over time, as much as 1.8 kilograms per doubling in total PFAS, as well as increases in hip girth as well, so body size overall. So now we have some evidence from prospective studies that PFAS are endocrine disruptors and obesogenic as well. One of the most consistent findings, at least in epidemiological literature, relates to PFAS and blood lipids. So this is meta-analysis results from multiple studies where they ask the question, what is the association between at least the two most commonly measured PFASs, PFOA and PFOS, and blood lipids. So PFOA has been consistently associated with greater total cholesterol, triglycerides, and LDL, which we know are detrimental for cardiometabolic health, as well as PFOS, with the consistent finding being total cholesterol and LDL, but not triglycerides. And these are a mix of prospective and case control studies. You can see heterogeneity on estimates. So this is why for the non-epidemiologists, it can be frustrating that sometimes there's a study showing a beneficial association, sometimes there's studies showing detrimental associations, but this is where we really need nuance and nitty-gritty detail on the methods to try to understand where biases might arise. But sometimes this meta-analysis cannot overcome this by pulling multiple estimates from different studies. Some of these biases average out, and you can obtain some sort of estimate of the potential effect. And I do want to point out, I put those response curves here, estimated from all the studies for PFOA at least, and you can see that these associations are fairly subtle, right? So they're not by any mean large, at least in terms of impact on lipids. But this is one of the most consistent findings, at least from the epidemiological literature that we have seen. So something that regulators and multiple agencies across the world, not only that, but also in the US, have used as an endpoint is immune dysregulation. And this is one of the first studies in the Faroe Islands conducted by Philippe Grandjean, showing that both PFOS and PFOA were immunotoxic. So what you're seeing here are those response curves, or PFASs on the x-axis, and then on the y-axis you have antibody response. And this specifically is a diphtheria antibody on the y-axis and tetanus on the following panel over there. But what he was able to show is that prenatal PFOS, so this is maternal exposure to PFOS, was associated with a 39% decrease in diphtheria antibody production in a population that is virtually all vaccinated against Tdap. Exposure in childhood also appears to be strongly associated with decreased antibody production in response to vaccines. So at age 5, PFOS and PFOA was associated with decreased antibody production of tetanus and diphtheria antibodies. And then not only that, these are decreases in total antibodies, but the question was asked also, well, does it have any clinical implication? And at least age 5 exposures, so exposures during childhood, were associated with greater odds of falling below the clinically protected level, what we think is protective of these infections. So this is one example. This is back in 2012. Now, some of these results have been replicated across other studies and as well as experimental animal models showing decreased immune response, particularly to vaccination, both from prenatal exposures, but also postnatal childhood exposures. So now there have been multiple studies. This is a recent meta-analysis from 2023, which looked at 14 studies. 13 of them were unique. There were 2 studies from the same population in the Faroe Islands. But you can see the estimates for all the studies there on the y-axis, and then this is a meta-analysis, with the null showing at 0. But what they were able to document, at least for PFOA, was a 5% decrease in total antibody types. So not only looking at Tdap, or the Tdap vaccine, but they also looked at things like HB, the influenza vaccine, and other common childhood vaccinations as well. And that was also present for PFOS, but the association was a little bit less robust. What has been very consistent in the literature is that both diphtheria, rubella, and tetanus has been the vaccine that's most affected by PFAS exposure. However, in the literature, we have only looked at 5 PFASs so fast. I mean, 6 at most so far. So it's definitely a need to try to get at more different types of PFASs as well. So what is our center doing? And I'm sorry, I don't have data to show you. We have partial data on the epigenetic endpoints, but the project 2, which I'm leading, we're looking at first trimester PFAS and metals mixture. So we're looking at the 6 commonly measured PFASs in plasma on the first trimester of pregnancy, as well as metal exposure, hypothesizing potential interaction. And then we want to explain the relationship that has been shown with prenatal PFAS and immune response by programming of leukocytes through epigenetic programming, specifically methylation of DNA. But we're also looking at cytokines secreted from children. So how is the immune system being programmed to hamper the immune response to childhood vaccinations? And one strength of our study that we'll be able to control for postnatal exposures as well, both metals and PFAS. So hopefully we'll be able to obtain unbiased estimates of the potential effect of prenatal exposure, but also the effect of postnatal exposure, accounting for prenatal. And we're measuring Tdap antibodies, but we're also measuring MMR, measles, mumps, and rubella, which are surgeons are coming back in some parts of the world. So I want to highlight, we're going to talk about regulation in a little bit, and it's really interesting. States are doing different things. Our local government is doing different things, but PFAS have no boundaries. So this is just a map of PFAS detection in drinking water. So we know that there's transport now across the globe, and water contamination affects multiple countries and states. So it seems silly to have this kind of state by state or even nation by nation set of regulation, where we really have to think about a comprehensive response, not only among individual countries or municipalities. So with that, I think I'll leave that for discussion at the end, but I just want to emphasize that we have vulnerable populations, and I think both pregnancy and early life are vulnerable periods to study PFAS toxicity, at least to set regulations. And then, we keep going back and forth about endpoints. So immune dysregulation has been proposed as a potential endpoint given the data, but there's also cancer, although there's not that much human data yet for the other PFASs. But, what about endocrine disruption and birth outcomes, which Amy will mention, which might be able to serve for risk estimates. And then we really need high quality. That's one of the things, prospective epidemiological studies, in order to provide more unbiased estimates of potential associations. And then I think this is a big topic for the larger group, but this issue of risk assessment based on a single chemical approach is really not scalable for the problem of PFAS. So we really have to think about this class approach that hopefully we'll discuss. So thank you for your time, and I think now we'll have Amy. So I think you're right there, and we'll pull up your slides, right? Thank you.